describing and predicting from transient patterns of brain function using hidden Markov models.
Thank you for the introduction. Great, my name is Christina Ahrens. I am a postdoc at Diego Vidora's lab at Aarhus University. And I will be talking about hidden Markov models and specifically how we can use them to describe transient patterns of brain function and how we can predict from them. Yes, right. So as we have already talked about a little bit, the brain is not a static object, but it changes its function over multiple spatial and temporal scales. And what I focus on is fluctuations on the level of milliseconds to seconds on the level of the whole brain. And I'm specifically interested in the patterns in which these changes over time happen. And the reason why I'm interested in this is that these patterns may actually be linked to aspects of behavior such as cognition, diseases like depression or consciousness. And the big question I work on, which I will come back to at the very end of my talk, is how can we predict aspects of behavior, disease and consciousness from the patterns in which brain function changes over time? But if that is the goal, if we want to do that, we first need to ask another question, which is how can we even describe these patterns? One way that we can look at how these temporal changes in the whole brain happen is we can do fMRI recordings. And I'm mostly going to be talking about fMRI studies. There will also be a little bit of MEG, which we heard about earlier. So if we do fMRI, we're not really looking at this like fast neuronal spiking, but we look at very slow fluctuations that happen on the level of seconds and that we can measure by the blood oxygen level dependent signal or the BOLD signal. And the idea is that if there's a lot of oxygen transported in the blood to certain brain areas, that means that probably in that brain area, the neurons need a lot of energy because they are being active. But it's kind of a very indirect measure and is a very slow measure. But this blood oxygen level dependent signal that spontaneously fluctuates even when someone is lying in an MRI scanner not doing anything at all. And we have also talked a little bit earlier about functional connectivity. I'm going to redefine it here because I mean something different when I say functional connectivity. In fMRI, because we are looking at these slow fluctuations, we just very generally mean that some brain areas temporarily synchronize their fluctuations, which leads to co-activation patterns. And that is usually what we mean with functional connectivity in fMRI. And there's evidence that just like BOLD amplitude, functional connectivity also fluctuates dynamically. So there are areas in the brain that are synchronizing and desynchronizing over time, which I here refer to as dynamic or time-varying functional connectivity. And I will say a little bit more about what I mean with that in practical terms. So let's say, so something that is more common to do is static functional connectivity or time-averaged functional connectivity. What people would usually do is they would take a time series like here, and the simplest case of computing static FC would be to just correlate each pair of these regions here in the entire time series, and then you get this matrix of the correlations between each pair of regions. And you can also project that into the brain, so you get these like connection strengths between your edges. And the idea is very simple, very similar to when we are doing time-varying functional connectivity. So if we are interested in how this functional connectivity changes over time, one way we can look at that is to do a continuously varying estimation in which we would, for example, take windows that we slide through the whole time course. And then we just look at the correlation within each window. So we capture sort of how functional connectivity evolves over that time course. Another way of doing that is with state-based approaches. And those are models that aim at describing this time series as a sequence of functional connectivity states. So what we would do there is we find time points that look similar, and then we summarize their description as states. So then each state would be defined by a functional connectivity matrix. And compared to this continuous estimation, one advantage is that you'd have more time points available for the estimation of each state. So they're more stable in that sense. So there are many different methods and many more complex methods and properly dynamic methods, generative models as well that I will be talking about generative models, but not the very complex ones. If you are interested in this kind of stuff, we have recently written a very hands-on introduction to dynamic functional connectivity with lots of little code examples. So if you want to try some of these methods out or compare them, or I think I said I was going to do like null models and simulations, which actually I ended up not putting in here, but it's in that preprint, which will be a chapter in like a methods book. But the model that I use the most is a state-based generative model called Hidden Markov Model, and I will talk about this model in more detail. I'm going to give you my my argument why I think that this model is particularly good, why I like to use it, and that is that being a state-based model, it is able to reduce these very complex brain dynamics into a subset of patterns of transitions between states, which we can actually describe statistically. And we can, for example, compare them between groups of subjects or something like that, or use it to study conditions in an experiment. And although more complex models, for example, higher order multivariate autoregressive models may explain the data just as well or better in some cases, these types of models are more readily interpretable. So we can, for example, understand what happens and when in response to a stimulus. And compared to other state-based estimators like clustering approaches, the HMM takes the order of a time series into account. But I'm going to say that the HMM is not a biophysical model like some of the ones that we have heard about earlier, but it is a useful description of the data. So what is the Hidden Markov model? In general, the HMM is a probabilistic model that assumes that observations, like it could be MEG or fMRI recordings, but it could be any other type of time series, were generated by a set of hidden states. And these states might, for example, be amplitude or functional connectivity networks. How does this then work? So we would start with a time series, for example, this bold signal that fluctuates in different brain areas, or electrophysiological data that fluctuates over time. And then the HMM estimates probabilistically from the data, both when states are visited and their spatial map. So for example, it could be a functional connectivity state that looks like this, and then some other number of states that have a probability of being active, or having given rise to the time series at each time point, and also a spatial map. And the HMM also estimates the transition probabilities between these states. And we can then use an algorithm to get a sequence of states from these probabilities, which is just a simplification that's easier to work with. Okay, you might, yes, sorry.
Does the model identify these states, or how do you separate the activity there into different states?
You mean so this is from the probability to the discrete state sequence, you mean? Or
I mean, or maybe I'm completely not understanding, but how do you partition these this activity that you see here, this measurement, the recordings that you see here, how do you partition them into different states?
Yeah, so the model estimates from the data the probability of each state being active, and then what that state looks like. So it's like an iterative inference process.
But like what?
Yeah, but what defines the state? What distinguishes a state from another state? I don't understand.
Because I think earlier in an earlier slide, you were saying you're kind of like trying to look for states that look like similar, and then you say this is state 1, state 2, like here, these colors.
Yeah.
So how do you know if like two sequences or two like snippets are similar to bin them into the same state?
I'm not sure I understand the question because, so you go through the time series and you look for time points that look similar. So you initialize the model somewhere randomly, the states and everything, and then you kind of, you try and find time points that look similar.
Yeah, then maybe my question is, what does look similar? Can I finish the question?
It's by the functional connectivity matrix, right? Like you had to figure.
So the matrices are similar by some.
The one before.
Yes, I'm going to come to that later actually. So what are states? Yeah, okay, that would be my next slide.
What do you mean by similar? What is the measure that you use?
Okay, sorry, I didn't understand. Yeah, that's my next slide.
Because I guess you have to predefine the number of states that you have. That needs to be predefined, but then the model finds what pattern corresponds to each state.
Yes, exactly. So I will say, like in this case, I have to specify there are 5 states or 10 states or however many. And then the model finds 5 patterns that look similar. And what these states are, so that would be, yes. So what are HMM states? I didn't understand the, sorry, yes. So the Hidden Markov Model is actually a class of models, and that offers a certain flexibility in assumptions. So you can actually say yourself what states are and what their distribution might be. So if you tell me that I don't believe in functional connectivity, I don't think that's a thing, then your states don't have to be defined in terms of functional connectivity. You can look for recurring patterns of amplitude only, or frequency, or whatever it is that you're interested in. So some things that people have done and that we've implemented are states can be amplitude states. They can have this like functional connectivity, some form of correlation or covariance. They can be phase coupling states, and they can also be frequency states. And it's always and or, so you can have states that are defined in terms of all of these things, where you go through the time series looking for similar patterns in terms of amplitude, functional connectivity, or phase coupling and frequency.
Thanks.
Okay, I'm going to show some examples, might make more sense then. What I wanted to say is that crucially, using something like the HMM and these different varieties of the HMM family, we can do this even for large data sets using a stochastic inference scheme in a single model directly from the raw broadband signal. So very briefly, I want to show you 2 examples of how this has been done. It has been, the Gaussian HMM has been applied to fMRI data, where states were estimated to have a mean activation and functional connectivity. So what you can see in the, you see my cursor? No.
Yes.
Yes, okay, but I can't. Oh, there it is. Okay, so we're going to do this. That's right. Okay. Aha, okay. So what you can here see would be one state that has a high probability of occurring at these red time points. And its mean activation map looks something like this. And then it also has an associated functional connectivity map that looks like this. And then you would have another state that has a mean activation map that looks like this and a functional connectivity matrix like this. And that occurs at different time points. And so when they applied this method to this large fMRI data set, where people were not doing anything, just at rest, they actually found these canonical resting state networks, such as which is shown here, the default mode network, or states that looked like these canonical resting state networks, or the language network, a visual network here. And they also showed that the transitions between these states are not random, but that they are actually organized in a very specific way. So here you can see the transition probability matrix. And you can see that there's these two kind of clusters. So it's again drawn here in a bit of a different way. So you can see that the transitions between states happen more within one group. So you have the blue group of states and the red group of states. And the brains are more likely to transition between states within that group than across groups. Yes.
Sorry, but and then you call the states language state, visual state, because they are correlated with the behavior at that moment of the.
No, I think so. There are these canonical resting state networks that lots of people have found in lots of different data sets. So kind of activation patterns that always look similar across data sets when people are at rest. And so what they did here was they took the map of these states and they just correlated the spatial maps with these established canonical resting state networks.
I don't understand why it's called language or visual state.
Oh, well, the original, that's how the canonical resting state networks are called, but yes, the original naming of those networks comes from some tasks. So visual network would be involving areas. You can see the occipital areas here. So visual network would be an activation map that involves a lot of areas that would also be active during a visual task.
Okay.
But here they were not doing a task, but these are networks that we know to also occur when people are not doing a task and they are just resting. The brain kind of goes through those networks regardless.
Okay, yeah, very clear. Thanks.
Okay. Another variety of the HMM is the time delay embedded HMM, and that can be used to detect frequency-specific power and phase coupling networks. And this method has been applied to resting state MEG data, here of 55 participants. And they found power and phase coupling networks, including sensory, motor, and higher order cognitive networks that again resemble these canonical resting state networks. So that's just to show that this method can pick out networks that we know from other data sets and from other models. So here, sorry, it's a bit difficult to see. Each column is one state, yeah? And the first one here would be this anterior higher order cognitive state. So you have the power map, the phase coupling profile, also the phase coupling profile here, and then a posterior higher order cognitive state. Then you have this sensory motor state, again defined based on the map, and some visual state again. And what's interesting about this is that if you look at the temporal characteristics, these states switch at very fast timescales, so between 50 and 100 milliseconds. And if we zoom into the first two states here, we can find that they look similar to these partitions of the default mode network that we might find in other data. So you have some anterior kind of portion of that default mode network that has a specific frequency profile, and the more posterior subdivision that also has its own distinct time scale. So our question was, how can we describe the patterns in which brain function changes over time? One way to do this is the Hidden Markov Model, which allows describing states of transient patterns of brain functions, such as amplitude and functional connectivity from MEG or fMRI data. And I also showed this time delay embedded HMM variety, which can be used to detect frequency-specific power and phase coupling networks at fast time scales directly from the raw broadband MEG signal. There are several papers where these different varieties of the HMM have been demonstrated, also in different modalities. And everything, all the code is available as a toolbox in MATLAB, and soon also Python, which we are working on right now. Okay, so this was our question so far. But the real practical question might be, how can I make sure that this actually works in my data? Because a lot of the time, these models are evaluated in these perfect, clean data sets, and then you apply it to a real study that has only 20 subjects, and the data is noisy and it doesn't work. So there are several practical considerations, and we have also been thinking a lot about reproducibility and reliability of these models. That's an issue that is not unique to the Hidden Markov model, but will apply to many of these models. So one thing to consider is that even when working with the same data sets, the result of one analysis might differ a lot depending on things like the pre-processing, sampling, parcellations, and so on. And the HMM is mostly data-driven, but certain parameters need to be set ad hoc, and that can also affect the results. Just briefly, what do I mean with pre-processing and parcellation? So fMRI data especially is usually way too noisy to analyze it as it is, so we need to pre-process it to clean it up. One aspect we need to think about is spatial pre-processing versus temporal pre-processing. And as you can imagine, if we want to look at some kind of temporal changes, temporal pre-processing is going to be really important, because otherwise you might pick up that one state is how much the person moves in the scanner. Another aspect is how aggressive the cleanup should be. So for example, motion scrubbing is something that some people like to do, but then, so you might want to get as clean as possible data, like squeaky clean data, but then you might also be losing a lot of variance that is actually important for the model to estimate something. And then we have talked a little bit about parcellations. So parcellations is just a term for how we divide the brain into areas. There are different sort of schools of thought on how that should be done. One question is that, should that be done based on anatomy? So like the actual folds of the brain, or based on brain function? Because we also know that certain areas kind of always activate together. And then there are these a priori parcellations, so like existing atlases that you can use to parcellate the brain. But there are also approaches that estimate the parcellation from the data. That's what I've illustrated here, where you would take functional brain scans of some group of people, and you could do, for example, an ICA decomposition. And then that would give you these kind of functionally relevant clusters of that whole group of subjects. And you can use that as a parcellation then. But that also means that one node might not be, the anterior, temporal, whatever, but it might be, one node might be already a network that you found across the brain. So it's a little bit trickier to interpret in that way. Yes.
And then do I understand correctly that if this experiment were to be performed again and the parcellation performed again, the partitions may not be the same?
Yeah.
Okay.
So the question was, how can I make sure that this model works in my data? So the problem that I'm referring to is the problem of detecting temporal changes, especially temporal changes in functional connectivity. So often we want to estimate time-varying functional connectivity in a group of subjects, to be able to compare, for example, how much a certain state occurs in a subset of subjects. So we start by concatenating that time series, and then we hope that the model finds something like this, where we have, again, 5 states that nicely recur across the group of subjects. So here, blue might be a visual state that occurs in each subject at different time points, and green might be the default mode network or whatever. And if we have something like that, we could look, for example, at how much time each subject spends in these different states, and whether that might be related to our research question. But sometimes this happens instead. So the model estimates that the best description of the data is that the whole time series of each subject is generated by one state. And there's several problems with that, one being that the states are not comparable between subjects. And more importantly, we expect the model to tell us something about temporal changes, but it's actually static. In practice, this problem occurs more often in fMRI data because of the higher spatial and lower temporal resolution, but it can also happen in MEG depending on the type of HMM. And I refer to this as model stasis because I didn't find a better word for it, but like a model that should be dynamic or time varying becoming static. We had several ideas of why this might happen. The first thing we looked at is variability, because we thought that maybe if subjects are very different from each other and there is little temporal variability, maybe static FC is in fact the best description of the data. We tested this in simulations. I'm here showing the mean measure of how static the model is as a function of the simulated variability. And in yellow is the area where the model does not find dynamics, and in blue, the area where it does find dynamics. And we found that when subjects are very different from each other, and when there's very little temporal variability, so this corner over here, that is where the model fails to find temporal changes. We were also interested in the effect of the parcellation. So here we used real data. We used the Human Connectome Project's resting state fMRI data set with 1,000 subjects. And so here we took the same data, but just in different parcellations. And this plot shows, so each color in this plot is a different parcellation. And we found that parcellation affects both how similar subjects are in terms of their time averaged functional connectivity, and also how the model is able to detect dynamics. And in fact, the parcellations where the model is most able to detect temporal changes are here, these group ICA, so the data-driven functional parcellations. Profuma is also some type of that. While the anatomical parcellation here in blue and the a priori defined functional parcellation performed much worse. And finally, here we looked at how changing the number of free parameters, sorry, about the parcellations.
And how do you define the model being static? So I get that if it's completely like separate, then sure, that's fine. But what if, you know, one of them is completely separate and the other two are sort of mixed? Is that bad or good?
So we use the mean maximum fractional occupancy as a measure. So fractional occupancy being the portion of the time series that is occupied by each state. And so the maximum would be the state that occupies the most or the largest portion of the time series. And if that is close to 1 over the number of states, then that would mean each state occupies the same amount of the time series. And if it's close to 1, that means that state occupies almost the whole time series. So the maximum of that, and then just the mean over all subjects.
Thank you.
Okay, so the free parameters and the number of observations, that obviously, I guess, also affects model stasis. And that is a simple statistical issue that if you ask any model to estimate many free parameters, but then you don't give it enough data to do that, it is just going to make a poor estimation. But that is very practical concern in a lot of these neuroimaging studies. We tested that both in simulations and in real data and found the same thing, that if you increase the number of free parameters, so this was the same plot as I showed before, but then we just give it basically a parcellation with more regions. So each state has then kind of a bigger functional connectivity matrix to be filled out. This yellow area where the model is not able to detect temporal changes grows. And if additionally, then we give it fewer subjects to make that estimation, this yellow area grows even further. So how can we make sure that this model actually works in the data, that it detects dynamics? we have, based on this, come up with some very practical recommendations. In the pre-processing, you should probably make sure that you reduce artifactual between subject variability, meaning make sure that your registration is really good, and preserve non-artifactual temporal variability. So as I said, you do want to do some good temporal cleanup to remove, for example, motion effects, but also if you remove too much of actual temporal variability, there are no temporal changes to be picked up. We also recommend using these data-driven functional postulations rather than a priori defined functional or anatomical postulations. And unfortunately, it is a good idea to keep the number of free parameters low, especially when the number of observations is limited, which is usually the case in a lot of kind of real-world studies, which means that you might, for example, have to give up some spatial resolution, so use a postulation that is not as fine-grained, or then do something like PCA, which, however, can affect the model in other ways. All right, again, so this is the reference to the paper and the code if you want to try some of this on your own data. Okay, now that this is done, we can move on to my big question from the beginning. How can we predict aspects of behavior, disease, and consciousness from the pattern in which brain function changes over time? And I'm just going to say I do not have the answer to that question, but I have some suggestions for you now. So it was actually recently shown that time-varying functional connectivity can outperform time-average functional connectivity in predicting certain phenotypes. So it is interesting to do this. And the problem is, though, say we have some model of brain dynamics like the HMM from a group of subjects, and then we want to use this model to predict something about the individuals who this model was fit on, such as their age. Now, the problem is really that it is unclear how best to use a model of time-varying FC or amplitude for prediction, because they're not as straightforward as static measures. So, well, the reason being that you have a lot of parameters. It's very high dimensional, so you have these like spatial and temporal descriptions. That's not easy to work with. So in the case of the HMM, you have these state probabilities. You have also transition probabilities. I'm gonna give them all like a parameter name. And then you also have the state parameters for each state. And of course, it would be possible to select one measure and say, I believe that it should be the occurrence of that particular state that is predictive of what I'm trying to do. But then you also lose a lot of information, and you do not capture the whole kind of description of these temporal patterns. And to complicate it even further is that these parameters all have specific relations with each other, and they are also on different scales. So these parameters, if we put them all together, I'm going to call them theta, they actually define a Riemannian manifold. And what we can do is we can construct a kernel, and we're going to use a projection on the Riemannian manifold to do that. I think I'm going to skip this. I'm just going to assume that people know what kernels are. So kernels are kind of a neat way to find a non-linear decision boundary using essentially linear models. And they're similarity functions. They're based on some kind of projection into a different space. So the general approach of constructing kernels from HMMs is that, again, we have some, oh, it's very small, huh? We have some neuroimaging time series from a group of subjects. We estimate the HMM on their concatenated time series so that we get the group level HMM parameters, so the state probabilities, transition probabilities, and the state parameters. And then we do a dual estimation where we basically fit this group level model again to the individual time series to get the individual, the subject level HMM parameters. And then we map each subject's time series, so each example, into a feature space and construct a kernel. And there are different options to do that. I'm going to zoom in a bit so you can see it. So we construct some kernel, and then we do a kernel-based prediction of some variables. And this is kind of this projection into a feature space, and constructing the kernel is kind of the core part to this. So let's look at this one. Option one would be to say we have in green, these green dots would be the subject level HMM parameters. And we could say we just ignore the fact that they lie on some Riemannian manifold, and we just look at Euclidean distances between those HMM parameters and construct a kernel from that, like a linear kernel. This is what I refer to as the naive kernel. We did actually try this to compare it to our approach. And this approach, we didn't come up with that, right? This is a thing from the 90s. It's called the Fisher kernel, which basically, on this Riemannian manifold, you have the group level parameters in gray here. And then you look at how for each subject, you have to change those group level parameters. So you're looking at the direction of steepest descent, and that is in this gradient space, which is a projection on the Riemannian manifold. And then you can look at the distances in this gradient space. Okay, so this would actually respect the structure of the hidden Markov model that this comes from. Here's the math. Some people here like math. You start by computing the Fisher score, which is the gradient of the log likelihood with respect to each model parameter, which basically just says how we would have to change the group level model to better explain a particular subject's time series. Okay. And then the practical Fisher kernel, I'm not sure if anyone. Okay, so we're using the practical Fisher kernel, which is simply the inner product for a pair of subjects. It's the inner product of their Fisher scores. We're also using a Gaussian version of this Fisher kernel, where we simply plug it into this radial basis function, and we have this extra parameter tau here, which is the width of the Gaussian kernel. Is this roughly clear? Did I go over this too quickly? That's very good, very good. So we did a study with the goal to find the optimal kernel to predict individual traits from an HMM. And the optimal kernel, we thought, should, well, first of all, be accurate, but also it should be reliable. And we defined reliability as it should have a low risk of making excessive errors, and it should also be robust to changes in training set. And we hypothesized that the Fisher kernel would outperform these naive kernels that are based on Euclidean distance and do not respect this geometry. And we also wanted to compare it to a previously used baseline model based on Kullback-Leibler divergence, symmetrized Kullback-Leibler divergence. We did this again in the Human Connectome project. This is maybe not that interesting. These are just details of the study. So basically, we have 6 different kernels in the end. So we have this naive kernel. Then we have just normalized it to see if that helps. And then we have the Fisher kernel, and then linear and Gaussian versions of each. And to assess robustness, we're doing a kernel-rich regression with each kernel. And then to assess robustness, we basically did 100 randomized iterations of 10-fold nested cross-validation to just see how susceptible is each kernel to being trained on a different combination of subjects. Here are the results. We found that across variables and these different cross-validation iterations, the Fisher kernel actually has a higher average prediction accuracy. So here I'm plotting on the left the correlation between the model predicted and the actual variables for all the different kernels. And on the right, this is the normalized root mean square error. So also a measure of how accurate the model is. And we found that in green here is the Fisher kernel, and we found that the linear Fisher kernel is actually the most accurate in both measures. And we also found that the linear Fisher kernel is more reliable. So here I'm plotting the maximum absolute error of each kernel, or the distribution of those. And you can see that here in green, again, the linear Fisher kernel, and then interestingly also the linear naive normalized kernel, they never get into this range where they make massive errors. But the Gaussian versions of the kernels, of all the kernels, and the Kullback-Leibler divergence, they get to the point where they are off by 1,000. And this is normalized, meaning that if you're predicting age, that model might predict that the person is like 1,000 years old, right? So that's bad. And then also in terms of robustness, again, the linear Fisher kernel and the linear naive normalized kernel did comparably well in terms of robustness. So this would be the standard deviation over the prediction accuracy for each variable. So we had these 100 runs of cross-validation, and ideally that shouldn't really change depending on what subjects you train your model on. And that is what we see in the linear Fisher kernel and linear naive normalized kernel. But again, you can see especially the Kullback-Leibler divergence-based model, it varies quite a lot depending on what subjects it has seen during training. This is just basically the same plot, but split up by variables. So the first one is age, and then we have a bunch of cognitive things that we predict. And in this plot, again, you can see in gray the Kullback-Leibler divergence, how far the spread is actually and how well it predicts. So depending on which subjects it encounters, sometimes it does a great prediction and sometimes it's very poor. And in green, the Fisher kernel, again, it's doing pretty well in the variables that can be predicted well. But of course, there are also some variables that are just not well predicted by any kernel, but that might mean that the information is just not in dynamics. So back to my initial question, how can we predict aspects of behavior, disease, and consciousness from the patterns in which brain function changes over time? I'm going to suggest that the Fisher kernel might be a useful starting point to do this, because it's a mathematically principled way of predicting from generative models, because it sort of naturally preserves the structure of that model. And I showed that it can be used to reliably make accurate predictions from a Hidden Markov model. And it can actually straightforwardly be implemented. So here I used kernel-ridge regression, but it's very straightforward to use it in a support vector machine or some kernel Fisher discriminant analysis, wherever your kernel method of choice is. And it can also easily be combined with other kernels. So we're actually working together with someone who combines sort of structural information, functional information, static functional, and then dynamic functional information in one model using multi-kernel learning. So or also information from different modalities. So that is actually quite neat. And I'm going to give you the reference to the preprint for this. And the code is also available also within the HMM toolbox. And that was it. Thank you very much for your attention. We are the analysis group at Aarhus University. Up here is Diego Vidora, and here's also my contact. Any questions? Yes.
Are you interested in interpretability of your model, or just in a prediction of the, I mean, you start with some data, which is fMRI, then you do whatever, but in the end, what you're doing is predicting the age or other properties of your patients. So my question is, are you only interested in the prediction and what happens in the middle is a black box, or are you interested also in interpretability of this prediction in terms of the brain dynamics?
So it is actually, that's exactly what I am planning to do next, because this was my frustration with this, that, okay, maybe now it predicts well, but having this kernel, you still essentially don't know where the difference between subjects comes from. So I'm actually thinking of taking one step back from the kernel and looking But looking at these different feature spaces, essentially, trying to predict from them so that you know what feature was predictive of something. Yeah.
Okay, so it's because you think the feature of this hidden Markov model are somehow nice in terms of interpretability of what's going on in the brain.
They might.
Yeah, it was just to understand. Yeah, thanks.
Well, thanks for the nice talk. I wonder if you can comment on the idea of states within states. So I imagine if you were to record EEG for a week, it would pull out sleep-wake. But within sleep, obviously, there's recurring states like REM and monitor. So are these methods advanced enough to pull out states within states, or are they influenced or biased by that potential? Or just comment generally on that.
So, I mean, you were talking about sleep. There's actually been application of the HMM to people sleeping in the scanner, and they picked up that there are states directly related to these like different sleep stages, and that how you kind of transition between sleep stages is then also linked to how you can transition between HMM states. So someone has actually done that. Otherwise, states within states. I'm not sure if this is what you mean with it, but there's I think he would be really mad if I call it an extension of the HMM, but that's how I understand it. But where basically instead of having at each time point one state, you have modes per time point. So you might be in like a mix of states at each time point.
Think of it as like maybe like a hierarchical cluster analysis.
Yeah. I mean, it's lots of, I've only talked about kind of the basic HMM where it's like you're in one state and now you're in the next state, which obviously is not how the brain works, right? We're not arguing that. But there's much more sophisticated models. But then also it becomes more and more difficult to actually fit that to data and more difficult to interpret it. But yeah, it's an interesting direction now.
Analysis now, is it on resting state data of it, or are there?
The last part that I showed?
Yeah.
Yeah, that was actually resting state data. What I've seen is that for this type of analysis, it's important to have a lot of data, a lot of time points per subject. And I imagine that the more, like the longer you see a person, the more you can kind of like have really individual parameters of what these temporal changes look like. And a lot of that is resting state data. I have done some like task data analysis with this. It's more difficult because of this temporal variance issue that I was talking about, and a lot of the time it's just more limited in task data for some reason. So I'm not looking at that as much.
Yeah, I'm trying to, going back to the interpretability, I'm trying to understand what could be the interpretation that you can gain from your analysis. So say you find that an older person, as I'm simplifying a lot, but has a higher probability of staying in the visual resting state rather than the language resting state. Okay, I find this, then what does this tell me about the brain function, or if it tells me anything at all? I'm not sure if that's where you want to go.
Yeah, I mean, so at this stage, the issue with the kernel is that it does not tell you that, right? It's because the input to the predictive model is just the kernel matrix. So the similarity of a subject that is old to a subject that is young. And so by that, I can't say if it's how much time they spend in the visual state or whatever else that made them look older. But if you take a step back and actually look at the features, then yes, I would argue that it is interpretable, and you could use it as a truly predictive model. So you could actually have brain scans of people. I mean, age, that's not an interesting prediction because we know usually how old people are. But if you were to apply it to diseases or something, or longitudinal data, and you see that if a person is lying in a scanner, they're not doing anything, if they then spend a lot of time in something like this default mode network, that is actually a very interesting state. That would be predictive of them developing Alzheimer's or whatever later on. That would be pretty interesting to know.
Yeah, so like a more direct
Yeah.
prediction from the features, you mean? Yeah, I see, thanks.
I was just curious, have you tried to also like look at the like similarities, like how did the HMM states compare to, for example, just doing like a PSA or something? Do you get similar like?
I think Diego has done that at some in one of the earlier papers where he kind of tried to establish the HMM as a model. It would be ICA or something like that, I imagine. Yeah. I have to look it up. I'm not sure now.
All right.
To follow up on this like states and states question, I mean, in your experience, how stable is the model with respect to choosing the number of states? I mean, if you choose 6 instead of 5, would you like see one state split, or does it like the topology look very different?
So, well, there's 2 answers to that question. The one thing is that it is related to how noisy your data is. So a lot of stuff that I talked about, if it's pre-processed well or not. So if the data itself is, if there are no obvious states, so obvious patterns that recur, the HMM is going to be more unstable as well. And then you might, even if you have the same number of states, get very different solutions because the model is stochastic. And then if you increase the number of states, well, anything could happen. If you have good, clean data and there are sort of obvious patterns that you could find in there, then it would be more stable. And something that then would happen is yes, that if you increase the number of states, you might get, for example, these subdivisions of some states. And actually, someone in our group is now working on, basically estimating how stable the HMM is in terms of if you just run it many times and then see how similar the solutions are, and then basically coming up with like a higher level solution where you only look at states that occur no matter how many states you estimate or no matter how often you initialize the HMM.
All right, then let's thank Christina.